So I will follow Mauro's presentation showing the efforts that we have done over the last year in the attempt to induce cardiac regeneration. And this is my only slide for introduction. Just to remind you that a major problem in current medicine is that cardiomyocytes cannot proliferate and regenerate the heart after any kind of damage. And this is because cardiomyocytes stop proliferating early after birth. So these are neonatal cardiomyocytes. As you can see here, you can see mitosis. They incorporate the nucleic acid analogs such as BRDU in their nuclei, as you can see here in pink, meaning that they can replicate their DNA and proliferate, whereas this is not possible for adult cardiomyocytes. And so one of the most important questions is, why do mammalian cardiomyocytes stop proliferating? Sorry, there is a delay in stop proliferating early after birth. There might be an intrinsic genetic control that forces them stopping proliferating, or there might be an extrinsic control. So something might occur at birth that doesn't allow them to proliferate anymore. And we are trying understanding what can happen at birth that is responsible for this switch. A first hypothesis is that the heart at birth is exposed to a hyperoxic environment, and this hyperoxic shock may trigger a response inside the cardiomyocytes, stopping their proliferation. So the hypothesis is that during the intrauterine life, the cardiomyocytes are exposed to a low oxygen tension, and suddenly at birth they are exposed to atmospheric oxygen concentration, and this stops their proliferation. So this is an interesting hypothesis that can be easily addressed experimentally. And so in collaboration with Esham Sadek in the States, we performed these experiments in which we kept the animals, the pups, either in a hyperoxic environment or in a hypoxic environment to investigate whether cardiomyocytes can proliferate more or less. And the answer is this. So essentially, if you expose cardiomyocyte to hyperoxia, what you see is that you induce more DNA damage, whereas if you keep the pups in a hypoxic environment, you protect them from DNA damage, and this results respectively in a decrease or increase in proliferation. So the overall conclusion of this story is that at birth, indeed, exposure to higher oxygen concentration induces a DNA damage response in cardiomyocytes, and this in turn blunt their proliferative capacity. So this could be one of the reasons why they stop proliferating. Another reason might reside in the sudden lack of exposure to maternal factors, which also occur at birth. And so a student in our laboratory performed an experiment by harvesting the sera from neonatal, pregnant, and adult animals with the hypothesis that during pregnancy, the cardiomyocytes could be exposed to some pro-proliferative factors, and that this stimulus could be absent after delivery. And so in this case, you should expect a capacity of the serum from pregnant animals to increase cardiomyocyte proliferation. And she collected the sera and she added two neonatal rat cardiomyocytes that can be cultured and kept in plates for days. So here, these are beautiful pictures of cardiomyocytes that you can see here in green because they express alpha-actinin, a marker for cardiomyocytes. And then we added EDU as a analog of a nucleotide, uridine, that can mark proliferating nuclei. So here in red you see the nuclei of cardiomyocyte that proliferate. So normally, if you add a serum from an adult animal, you have like 10% of proliferating cardiomyocytes, but if you treat them with a serum harvested from a pregnant animal, this increases up to more than 20%. So there is something during pregnancy that increases cardiomyocyte proliferation. And we reason that one of the cell population that are expanded during pregnancy to induce maternal tolerance toward the fetus are regulatory T cells, which are a subset of T cells that blunt the immune response against foreign antigens. And when we tried the effect of Treg condition medium on cardiomyocyte proliferation, we could reproduce what the serum from pregnant animal does. So one possibility is that Tregs act as the source of pro-proliferative signals that induce cardiomyocyte proliferation during pregnancy, and they are not present anymore after birth. And just to confirm this hypothesis, we depleted Treg in pregnant animals. And you can easily do this because we have an antibody against a marker of Tregs that is also functional in keeping Treg activity. And then administer BRDU to pregnant animals. And what we see from these results is that Treg depleted pregnant animals allow a lower cardiomyocyte proliferation in the developing embryos. And now we are trying to understand which are the factors, the soluble factors that are secreted by Tregs and can control cardiomyocyte proliferation during embryonic development. And we had the chance to perform a bioplex cytokine plex assay, assessing the expression of various cytokines. And just to make a long story short, we have identified a couple of cytokines that are highly secreted by Tregs and are also highly present in the sera collected from pregnant animals. So we are now exploring in more detail the effect of these cytokines on cardiomyocyte proliferation. And then beside these stories that try to understand why cardiomyocytes stop proliferating at birth, we also underwent an unbiased screening, asking ourselves whether microRNAs could be involved in controlling cardiomyocyte proliferation and thus used as therapeutic agents to promote regeneration after damage. I'm not going into the detail in the microRNA pathways, just to remember that we have about 2,000 genes encoding for microRNAs in our genome, and these small RNAs are transcribed by the cells and can match target messenger RNAs in the cell, overall inducing translational repression, so silencing the expression of hundreds of genes at the same time inside the cells. And you can deliver this microRNA to the cells either exogenously as microRNA mimic, so as double-stranded RNA, or you can clone the microRNA precursor into viral vectors that can be used to transduce your target cells to overexpress these microRNAs. So here at ICGEB, we have set up, as Mauro said before, a high-throughput screening facility that allow the automatic plating and dispensing of reagents into multi-well plates. And with this facility available, combined with a high-contact microscopy, we could seed neonatal cardiomyocytes in 96 well plates, and in each well we added a single microRNA out of the 2,000 microRNA in our genomes. Actually, initially we performed the screening with just the first 1,000 sequences that were known at that time. And then we added EDU, as I showed you before, to mark proliferating nuclei. And then we took the pictures and analyzed the amount and the percentage of cardiomyocytes proliferating in each condition. So again, this is the original microscopy. Again, you see here in green cardiomyocytes. In red, you have nuclei that have incorporated EDU, meaning that their cells are proliferating. And in blue, you have all nuclei. And this is the image acquired by the microscope and elaborated, and also automatically giving you the percentage of cardiomyocytes that carry an EDU positive nucleus, color here in yellow. So normally this percentage is about 12%, as I showed before, and we could identify about 40 microRNAs that significantly increase this percentage up to more or less 50%. And this works in both mouse and rat cardiomyocytes. And this is perhaps the best convincing picture to show you that these cardiomyocytes are actually proliferating, because if you leave them there for 6 days, they fill the plate. So the plate is full of proliferating cardiomyocytes. So what happens if you take some of these microRNAs and you inject them in vivo in a newborn heart? This is the results after a couple of days. You see that this is a control heart, and these are two hearts injected with the two of the most potent microRNAs, microRNA 519 and 119A. You see here that these walls are much thicker, and it's not very well visible here, but we also label proliferating nuclei in vivo. I show you an enlargement. You see that a lot of cardiomyocytes in green are actually proliferating. They incorporate EDU in vivo. So initially we tried the microRNA as a mimics, and then since we are in love with these viral vectors, the AAV vectors, we cloned the respective microRNA precursors to achieve long-term expression of these microRNAs in vivo. And these are the results. So if you inject the pups with an AAV vectors overexpressing miR-519 or miR-119A, you end up with larger heart. This is hyperplasia, so you have more cells. It's not hypertrophy because the single cell diameter is not increased. And this is a sort of a physiological hypertrophy because these hearts work perfectly. If you do an echocardiographic studies on this heart, the pump function of the heart is perfectly preserved. And again, if you look at the presence of mitosis, you can detect an increased number of cardiomyocytes that are still proliferating after birth. So they are undergoing karyokinesis and actual proliferation. Can we use these microRNA therapeutically? So to address this question, we injected the same AAV vectors encoding for the two proliferating microRNAs in a mouse model of myocardial infarction induced by the permanent ligation of the left descendant coronary artery. And these are the results. So here in blue, you have a staining for fibrotic tissue. You see that since a couple of days up to 2 months after the ligation of the coronary artery, you end up with the loss of a portion of a contractile heart, which is completely replaced by a fibrotic scar. Instead, if you deliver the two microRNAs by AAV vector, intracardiac gene transfer, you can end up with a reduced infarct size, preserved heart function. These are all parameters of cardiac function by echocardiography: ejection fraction, fractional shortening, and anterior wall thickness. And so the overall message is that these microRNAs can induce regeneration and preserve cardiac function after myocardial infarction. So this is nicely published, and now the challenge is to translate these results into a therapy that can be offered to cardiopathic patients. And so we are dealing with several issues that has to be resolved in order to achieve this goal, and I will show you some of the preliminary data that we obtained in these different fields. So first we have to understand which are the target cells that are responsible for the effect of the microRNA. So one of the most frequent questions that everybody asks us is, but do they act on resident cardiac stem cells, or they are able to induce the proliferation of adult cardiomyocytes? So to understand and resolve this issue, we set up a quite complex fate mapping approach, but basically we crossed mice that express a Cre recombinase, an inducible form of the Cre recombinase, with the reporter mouse that switch on GFP expression upon Cre recombination. So by administering tamoxifen that activate the Cre recombinase to these mice, we could switch expression of the GFP on only in cardiomyocytes, because the promoter driving the expression of this recombinase is a cardiomyocyte-specific promoter. So eventually, after 7 days of tamoxifen treatment, you end up with a green heart, in which the green cells are differentiated cardiomyocytes. So if you induce a myocardial infarction and then you administer your microRNA to this heart, and then you give the animals the EDU analog to mark proliferating cells, eventually you can understand whether proliferating cells arise from these green adult differentiated cardiomyocytes, or they do arise from some black cells that have not been marked, and so they were not already differentiated toward the cardiac lineage at this stage. And this is the result. So what we could see is that most of the EDU positive cells are indeed green, meaning that they derive from already differentiated adult cardiomyocytes, and this is perfectly consistent with the ability of these microRNAs to induce proliferation also from of adult cardiomyocytes in culture. So the data that I showed before were obtained in neonatal cardiomyocytes, but now we are also able to culture adult cardiomyocytes, and these microRNA are still working in these cells. Another important issue is to understand if they are also affecting in human cells, if we want to treat patients eventually. And so we pursued different complementary approaches. First, we had access to cardiomyocytes that can be derived from human embryonic stem cells. And we shortlisted 8 microRNAs that also induce the proliferation of these embryonic stem cell derived cardiomyocytes. In the meantime, a couple of students learned how to culture human cardiomyocytes, both from adult heart, from patients undergoing cardiac surgery and extracorporeal circulation, and also from human fetal tissues. And again, out of this short list of 8 microRNAs, we could identify a few ones that are particularly effective also in primary human cardiomyocytes, and so we are moving toward the experimentation in large animals using these selected candidate microRNAs. A last important issue is to optimize as a delivery route a formulation, because as we learned from the previous talk, going into therapy with the use of vectors is more and more difficult because of several problems that have arose over the last year. And so we started assessing whether we could deliver these microRNAs as synthetic molecules, synthetic RNA duplex, instead of using viral vectors. And quite unexpectedly, because we thought that these molecules were quite unstable in vivo. Instead, when we injected this double-stranded RNA molecule in vivo into the heart, we could detect their presence up to several days after delivery. And by extrapolating their persistency from this curve, we expect them to be present up to almost 2 weeks, which could be a reasonable time to induce a wave of proliferation and regeneration after an acute damage. And not only they are present, but they are also functional, because if we look at their main target gene by RT-PCR, we see that they are decreased, meaning that these microRNAs are functional and they are silencing their main target. And so this student, very talented medical student, repeated the expression of myocardial infarction therapy using the synthetic microRNA instead of AAV vectors. And as you can see from the graph, we could almost reproduce the results. They are a little bit less effective than the AAV vectors, but still the scar is very reduced, delivering synthetic microRNAs. Ejection fraction and fractional shortening are preserved, and we could detect proliferating cells. Another final issue, and then I'll stop, is understanding their mechanism of action. So what are the mechanisms by which they trigger proliferation? And to try to start addressing this question, we perform a transcriptomic analysis by deep sequencing of cardiomyocytes, either control cardiomyocytes or after treatment with the 2 microRNAs. And we identified almost 700 mRNAs that are downregulated by these microRNAs. And since in our facility we also had a library of siRNAs, what we could do was to silence one by one these 700 genes and see the effect on cardiomyocyte proliferation. And this is the result. So we could identify a couple of potential microRNA targets that, when silenced individually, are per se sufficient to increase cardiomyocyte proliferation over control, although, as you can see here, none of them is as effective as the microRNA to increase cardiomyocyte proliferation, meaning that probably cardiomyocyte proliferation relies on the contextual and simultaneous silencing of multiple target genes. And as I just want to drive your attention to the fact that several of these genes that are downregulated by microRNAs that increase cardiomyocyte proliferation are genes that are somehow part of the contractile apparatus or the cytoskeleton. So one of the ideas that we're pursuing is that the cardiomyocytes, in order to proliferate, have to disassemble somehow the cytoskeleton that can represent an inhibitory issue to the proliferation itself. So to try to reproduce and to deeply analyze this hypothesis, we try to silence multiple sarcomeric proteins that are known to be downregulated by the microRNAs in a combined manner, to see whether the combined silencing can mimic microRNA proliferation. And at least for some combination, I show you here this a combination of these 3 proteins of the sarcomere, titin, cofilin 2, and anchor D52, we could achieve a similar proliferation rate as driven by one of the microRNAs that I showed you before. So with this, I would stop. Of course, I have to thank you, all the people working in the molecular medicine lab, and in premise Mauro Giacca. And here you see some collaborators of the cardiology department in Trieste, and also the people working with us implementing large animal models of cardiovascular disorders. Thank you.